These cells can differentiate into cells of the respective lineages, meaning that the hematopoietic stem cells can produce all cells of the blood system. They can't produce other cell types. And if you go back through the literature and try to comb through this, I'll try to save you a lot of time from a lot of artifacts and things that came out of studies, because they've all pretty much been proven not to be true. And that is that hematopoietic or mesenchymal can turn into things that are not within their lineage. And this, again, I'm happy to discuss afterwards, but there's plenty of experimental systems that have shown that not to be the case. That being said, there's still tremendous utility in these cells. There's been a lot of information that we have learned from studying with these cells in various types of systems. And I'd say most importantly that these actually have the most current clinical utility. Some of these have been used already for clinical therapies for a long period of time, and others are well on their way. So there are also stem cell types that are constantly playing a role in terms of regenerating and the renewal of our tissues. And skeletal muscle is one. Every time you exercise, that you actually have regeneration that occurs as a result afterwards. When you work out, this is how you bulk up. The epidermal stem cells in our skin are one of these, and our intestinal system. So there's numerous ones that are highly specialized stem cells that can actually contribute to the tissues over the lifetime of the individual, versus the ones that come from, say, earlier stages that have greater potency that contribute more to the development of those tissues in the first place. So they play just different roles in different stages. So when stem cells were first being thought of, particularly for clinical types of therapies, there's two sort of routes that were intended or have been followed for this. The first of these is actually in cell transplantation. So the idea being that if you have stem cell types or cell types, they can be directly injected into tissues and used as a means to promote or to integrate and replace cells that are lost. And two of the issues that came up with this initially, and I'll discuss some of these and how the challenges, have been cell survival. So having healthy cells at the time of injection is great. Having them survive for minutes, hours, even days afterwards has been a real challenge. And also then tissue integration being the second part of it. So if you can get them to survive, do they actually ever contribute functionally to the tissue that you're trying to target them? This is still a challenge within the field. Secondary to that, many people have tried and attempted to actually then engineer tissues out of cell sources. And the idea being, again, that you can direct the differentiation to some lineage with a target tissue in mind. Challenges here, again, are oftentimes with regards to how do you make a tissue in the first place, particularly from stem cell sources, let alone a cell type that already has some directed differentiation towards it. So this has been sort of where I'd say the first generation technologies and where stem cells started to enter into the fields, particularly for regenerative medicine. Some of the challenges associated with that then, there are many of these, so I'm only going to highlight a couple, but immunology is one of these. And this isn't a new problem. This is the same problem that was encountered with organ transplantation in the first place. And that is that if you don't have patient-matched cells or a means by which you can try to evade the immunological system, then it's nearly impossible to try and get those cells to survive and persist and integrate and do the types of functional targets that were initially envisioned. Interestingly, though, there's also the fact that when you introduce cells like this, that there's always going to be biological crosstalk. And so there's actually one of the things that's come out of this is that there's a lot of immunomodulatory behaviors of various stem cell types, particularly mesenchymal stem cells have been one of the ones that have been most well studied in this regard. With regards to then trying to get the cells to survive, whether it's the cell transplants or whether it's actually the engineered tissues, that vascularization of the endogenous tissue has become, again, one of the limiting factors. So trying to engineer a vascular bed or system ex vivo, again, is when you think about it, a very difficult, if not almost impossible challenge to approach. And that is that even if you can get a vascular type system throughout a 3D network, how do you get that properly connected to the host vascular once you do? And this has been tried, again, I would say now over 20 plus years in tissue engineering, and there is still not a commonly accepted route by which that occurs. So it's clear from this, again, that there has to be something that from the vascularization that probably has to come from the host itself. And this, again, changes because if you're trying to get large tissues that are dependent on this, it's again, very challenging, if not impossible, to try and get that to occur right at the beginning. So it changes the paradigm in terms of how to try and attempt to grow tissues and integrate them in a functional and meaningful way. And then the functional tissue integration is another. It's one thing to have cells persist. There's a lot of imaging things you can see to say that cells are alive. They have a luciferase reporter activity. But in the case of, say, heart muscle, do they ever functionally couple to the heart muscle, the host cells? Because if they don't, what you're potentially introducing is a greater arrhythmogenic risk than what might already exist. Now, interestingly, there's been recent results actually from former lab where I was actually did my training as a postdoc that actually suggests that unexpectedly, and why this was made a lot of press, that the introduction of cells actually has anti-arrhythmogenic properties of certain cell types. Now why that is still to be defined, but that's an interesting find that had they not been doing those types of grafting studies in the first place, you might not have found. And it does say that several times many of these have to be, assumptions we make have to be challenged by true experiments to test these out and find out if that actually is the case or not. And if these kinds of more scientific side of things can be addressed or effectively solved, then one of the other things that still is behind all of this, perhaps, is the challenges associated with how are we going to make and produce these in a way that is not just commercially viable. That's one of the things, is to make these things so they don't cost such a ridiculous amount that they are never used for most types of interventions, but also how do we properly educate regulatory agencies about what's in the pipeline, what the intent of these are, and provide them with the proper information so that they can actually get these through to the clinic. Facilities and processes I'll touch on a little bit later, but this is the common one. I mean, it looks today nothing more than what our research labs look like. And yet if you go to any kind of manufacturing plant, say, to see how they produce cars, you see very few of these people. You see robots all over, you see assembly lines moving along, and the question is are we going to get to that kind of state with these types of technologies or not, and when would that occur? So what I'd then like to come back to and highlight is that there have been paradigm changes that have occurred in this field, and there's two of them that I'll touch on and kind of segue into some of the things we'll talk about today. The one of these is that with regards to cell transplantation, one of the things that became noticeable was that even in the cases where you didn't get cell survival and you didn't get tissue integration, there was some type of effects, transient as they might be, that could actually alter the trajectory of the wound healing response such that you ended up at a different outcome, and in many cases, or in some cases, a better outcome than you would have had there been no intervention. So this created then secondary hypotheses, one of these being what role would the cells be playing, and one of the most popular ones still currently, and has started to bear itself out with additional experiments, is that actually the cells serve as a complex drug delivery vehicle. That stem cells introduced, and perhaps it could be due to the fact that you're introducing in an injectable sense supraphysiologic levels. Maybe there aren't that many stem cells that exist, or not that many that would traffic there anyway, but you actually start to get increases perhaps in local factors that can alter the course of the tissue regeneration or wound healing response. The other is that in some cases, and this is oftentimes the simplest one, this has actually been shown that sometimes it doesn't matter what cell type you put in, but if you put in cells versus something at a higher abundance than would normally be there, and it would be acellular, that actually it provides mechanical stability and support to a tissue, and that can prevent subsequent deleterious events. So this is perhaps not as exciting, because then you say, well, this could be any cell type, and it's not so stem cell specific. But this first one here, particularly with the trophic delivery, has incited a lot of interest because it does seem to be more specific, or there's something different about stem cells than any old cell. The secondary thing is that the thoughts about, well, if all of those challenges I mentioned about trying to get tissues to integrate into an in vivo tissue exist, then what other opportunities are there? How far off are we? And one of the ones I'll just touch on is the opportunities to think about smaller size tissues that model or I would call them units, functional units or components of larger tissues that can function with something on the multicellular scale that you wouldn't get out of an individual cell alone. Meaning that an individual cell plucked out of this might give you some information about the transcriptome, the genome, and other things, but it doesn't tell you about the cell and how it's communicating with its neighbors, how it's functionally integrated within that. And this is, I think, potentially a very powerful platform for a lot of things that can be done in vitro. In particular, drug screening is one of those, and we'll hear some of those types of talks today. So what are the remaining challenges? We're not done yet with this. I would say a couple that have changed the landscape but still require a lot of work. Reprogramming is the hottest and newest thing in the recent, now just slightly over 5 years. But we still really don't understand this is what's going on. This is still largely a black box. So the processes from which reprogramming starts to where it ends is still, it's given names like Area 51 and other things simply because it's still a ill-defined process, and we don't know. We know what the result can look like, but we don't know necessarily how it occurs. And if we don't know that, then the questions about efficiency, reproducibility, how do you use this in a more targeted way sometimes can be limited. That being said, I mean, this has become a very robust technology, and we'll hear this from a couple of speakers today, in particular Steve Stice, who has a lot of experience with this. one of the challenges I see is that if you can reprogram cells to various states, one of the things is if you want to get them to go back to a more differentiated state. So this opens up possibilities for making a huge array of different, or having a huge diversity in terms of the genetic information available for various types of studies. What it doesn't do, though, is it doesn't necessarily get us back to the cell types of interest for certain types of applications or tissues. And so this is still something that, while there's been tremendous progress, and again, we're going to have two, I think, excellent talks this morning about people who have made significant progress for specific cell types in this arena, this is not translated that these are things that just any old lab can do at a whim. These are not kits that you can buy. They're still require a fair amount of work to be able to have an array of all the various cell types that might be of interest to researchers and to clinicians. And then the other part of this is also paracrine effects, and we'll hear some of this again through some of Jana's talk, about what is going on in these complex environments. Can you start to dissect this, and can we start to understand how the environment actually plays some role in this, and how is it that cells are responding to the environment and other cell types with these molecular secretion dynamics? If we know something like that, then going back to the trophic delivery can become a very targeted and directed therapy, as opposed to right now where it's uncontrolled or it's at least ill-defined what's going on. So let me just touch on this briefly by way of introduction, and Steve, I don't know if I'm using anything that you have in your talks with this, but I wanted to at least mention induced pluripotency as one of these. The ability to reprogram cells has occupied numerous reviews, but it's also occupied an amount of research over the past, like I said, 6 years. Almost everyone, or many people working in stem cells, those particularly in the pluripotent side of things, have either gotten into reprogramming or are using it now, I would say, routinely as a tool in the context of their studies. And the reasons for this is that while this number sounds very low in terms of efficiency, if you use, for example, hundreds of thousands of cells, and you get even a small percentage like this, which accounts for a couple of cells, which you can then selectively expand, you can pretty readily start to think about how you can have iPS cells, at least, working within your lab within perhaps a couple of months. That's the theoretical, working with those cells properly, knowing how to culture them and without them differentiating prematurely, things like that. That's a little bit more, requires a little bit more expertise. But at least, theoretically you can say, well, this is enough to functionally get started with this and to make this something that numerous companies are now selling reagents for. So you can pick up a number of different types of kits, be they viral-based, non-integrating, integrating, plasmid DNA. There's every potential variety out there. There are mRNA-based approaches. There are protein approaches that have been at least attempted. I don't think those are necessarily commercially available yet. And there's microRNA. So anything you can think of to manipulate at some level within a cell has been attempted and has largely been at least one published report that it can be used to induce reprogramming of the cells. There are now, and I saw this just actually from this review from George Daly, one of the leaders in this field, there's now more than 45 different types of diseases. So pretty quickly, in short order, we have a large number of human disease models, or at least the start of human disease models that are somewhat readily available. And this again means that using this technology, if you have a patient of interest, a population of interest, reprogramming enables you to now have an abundant source of those cells, because these cells can be expanded indefinitely under the appropriate conditions. So you don't have to go back and biopsy. You're not limited by what you obtain initially. If you can get reprogramming to occur with some of those cells initially, you have your stock that you can work from. And now you can also look at the diversity across populations. And this has already started to be attempted, and these are just some of the most common. These aren't all the ones, but many of the most common ones that now there are some of these differentiation protocols, or at least relatively routine, that labs are examining the differentiation of these cells, so that now in their culture dish, they're looking at basically something that they would normally only look at maybe by an epidemiological study. So this is quite transformative to the field. It's quite powerful, and it is radically and rapidly changing, I would say, how much of human biomedical sciences are going to be done in the future. So for that, and this is pretty short order, but this is yesterday's news, and so this is straight off of their website. But the two gentlemen, or the one gentleman who's credited with reprogramming in the current age, and one who many people have forgotten, but what he laid as the groundwork for this half of a century ago, was the demonstration that there's a tremendously more plasticity to cells and to the genetic information contained, or that the genetic information contained within cells that can be induced under particular conditions. So John Gurdon, who's famous for his studies in frog, this is where most of this was done, could demonstrate that you could transfer nuclei in and get reprogramming to occur. And Yamanaka did it the opposite way. If you take the factors into the differentiated cells that are from stem cells, then you can actually get it to occur from the inside out from the nuclei. So these two contributions, which I always also like to point out, this is from, I think it's Journal Developmental and Experimental Cell. This, by today's standards, not probably anyone in this room has this as their table of contents. No one reads these things, but if you go back to many of the landmark or seminal studies, they're oftentimes in these journals. Versus the ones that we frequently see, like Cell, Nature, and the rest of these, where most of the IPS major publications are typically found. So it just brings up 6 years is probably one of the most record times. I think that rivals perhaps prions in terms of from time of discovery to actual Nobel Prize. So I think it's been widely appreciated. So let me then, with just the remaining couple of minutes, okay, just a couple of quick minutes because I want to get us back on schedule. Just go over a little bit of some of the stem cell engineering things. And so I've talked about this in largely the biological sense, but I think one of the things that's important is this amount of basic science knowledge that has been acquired and accrued over the past several decades, perhaps 30 or so years. We're at this point where now these types of things, which are still largely cartoons in my talks and in most of the others, are actually at least closer on the horizon to becoming reality. But I still think that this is the realm in which, at least with an engineering approaches, and hence the stem cell engineering title, where we can start to think about how we can actually embed within this pipeline the robust types of approaches that make it possible for these to become efficient processes, that these can be things that are manufacturable, and these are commercially viable types of things. And this is occurring. I don't mean to say that in the absence of engineering this isn't occurring. It is. I just think that when we look at the parallels between various other types of fields, when you look at the basic science and you look at the applications, engineering has traditionally played this type of role to help facilitate this process. I think the other powerful thing is that there are a number of new technologies that can also be developed along the way. These feed back into the pipeline to hopefully enhance and improve the type of basic science and research that we're doing. So I'm going to skip a couple of slides, apologize, but like I said, since we started a few minutes late, I think it's important to give our speakers the full time that I had asked them to talk about it. But I'll mention just that this has been something that's on the radar of the federal agencies. So this past year and a half, and we just concluded, and Bob Neera mentioned this yesterday in his regenerative medicine talk, but Bob and I, along with 4 other folks, were fortunate to be a part of this study, which basically was to examine what is the role of stem cell engineering and is its development as a potential field. Numerous other studies like this have been done by this same group that's contracted by the government for things like nanotechnology, tissue engineering, things that now we appreciate and are widely known, that as they developed at a nascent stage, this was the type of study to try and identify what was going on early and what are the potential types of funding initiatives that can go along with that. There's a number of recommendations that came out of this. A couple of things is just where does this start in terms of the physical engineering principles and how does it influence the cells? There's a tremendous amount of technology development and how this can be playing into things. There's also an important role for computational modeling in this. This is most of the folks that are at least initially attracted into these fields, myself included, I have to admit, we're the soft and squishy engineers. We like cells and molecules and things like that. Oftentimes we are not as comfortable in front of computers and programming, and yet actually I have to admit that I've turned full circle on this as well, or 180 degrees. And I see now tremendous potential. If we can understand and develop some of the algorithms for this, this is a great opportunity for computational modeling to come in and really aid us in trying to survey this tremendous experimental space that even these types of technologies I don't think are fully going to be able to cover. And then all of this, how can we start to implement these types of principles and findings into the pipeline and the manufacturing side of things? The Stem Cell Engineering Center here, I'm just going to skip this. I think I've talked about it. Maybe I'd be happy to talk about before, but something that at least at Georgia Tech we appreciated how these things, even before this type of study, how can we play a role in this? There's tremendous engineers here, as along with our colleagues from UGA and Emory and in the local Atlanta area that are working on some of these issues, and we want to build a group and team together to address them. And I will skip these because of the time, so I apologize. But I want to highlight one thing that came out of this. This is actually technology because it does involve multiple people. This is actually primarily driven out of Andres Garcia's lab, and he mentioned, I don't think he has talked yet, so I don't think he's mentioned anything about this, but a technology that now there's been patents pending. This has been a great collaboration with Steve Stice and his group as well on this. and actually just recently now to expand this work with an SBIR with Steve's group. So one of the things with this is if we understand mechanistically more about the cell types and populations, in this case using adhesion properties, we can actually then go back and use simple microfluidic systems to be able to go through and actually isolate these cells in a much more defined way. Note it, the typical way to do this would either be in batch cultures where you use enzymes which are not selective, or again, the timing of things, you can get detachment of different cell types at various times. So in a dirty type of culture where some of these can sometimes start from either a reprogramming study, if you're culturing on feeder cells, or if you have some level of spontaneous or even directed differentiation going on, purification is always one of the sort of bottlenecks. In this case, within 10 minutes using controlled shear forces, you can actually detach cell populations of interest. You can do this in a highly efficient and with a high yield as well. So this has been extremely powerful. This doesn't change the cells at all. We've done a lot of work with this, and that's what most of this data shows, is that the cells going into the device and culture in the device are no different by a short-term exposure to shear. So I talked about physical effects, but this is something on the order of 10 minutes. This is sort of a refractile period where the cells don't change. So this has been something that we've looked at throughout, from early stages of being able to separate different cell populations out to later ones, and this is something we're continuing to work upon. But I'm just impressed that there are very few techniques that can get to this level of reproducibility and detachment. And this, again, was a great collaboration, like I said, from our stem cell biology principals and colleagues, along with engineers who, and Andres will readily admit and has said this, so I can say it for him, he would not have gotten into this type of work had it not been for the fact that there were groups and people now starting to work in human pluripotent in particular. It wasn't a cell type that he's ever worked with or published before, but having the capabilities and resources and knowing what some of the challenges were, this was a great opportunity for him to get involved with this. And I think that that's what I see, something that can continue to spread across Georgia Tech and the types of labs and investigators that we have here. I'll skip this as well because I just wanted to get to this slide. Steve Cross always talks grand challenges, so in the spirit of Steve, I will convey what I think are the grand challenges, or at least maybe the grand opportunities that I see in the near-term future with this. And there's three of them in particular. They're linked, so I've tried these divisions are maybe somewhat arbitrary. The first one is this term, and I know it was used even earlier today, predictive and personalized medicine, and what does that mean? From the outside, you know, I've oftentimes not understood exactly all of that or what some of those things are, and I've grown to appreciate and understand better. But when I think of IPS and what it represents, this is at least perhaps to me one of the most tangible examples of what could be personalized and predictive. So if you have cells that contain your own genetic information that can be examined in an in vitro setting where drug screening and other things can be done, I don't know how much more predictive you can get except for to orally take those drugs and see what happens. And again, I'm simplifying this, I recognize, but this is about as close as perhaps in the near-term future we're going to get, unless we actually did have some type of island of clones where we are using them for spare body parts and other screening ones. And I think that as much as that has been used in several science fiction books as a topic, it's not something that is really ever going to be something that occurs. But this is actually something that is quite manageable, quite attainable, if costs can be addressed. And that's, I think, the biggest challenge to this. But this has tremendous potential to me. The other one that I oftentimes think about this is that, you know, and this is an example of, say, twins. How is it that, you know, you can have the same genetic information, but it doesn't yield the same outcome over time? And we know quite simply that, you know, nature plays its role and there's a genetic code, but epigenetics overlays that, and there's a number of environmental influences throughout our life that shape and mold our health. And this is an opportunity, again, where we can actually start to think in a predictive or personalized fashion about, well, if you are exposed to these things or if you're predisposed to something, are there interventions that could be taken earlier, or are there certain types of things that could be avoided? Again, I realize that's pretty far off in a way. However, it's again something that if you put some of the pieces together and the logic of this, it's possible. We just need to think about how that might be attained. And the other one is that this is one I think, again, if you go back now, and I've oftentimes been surprised, we really still know quite little compared to all of the other organisms out there about our own development. We can take snapshots and pictures of this, but there, again, for obvious ethical types of reasons, there are no tractable systems whereby we routinely do experiments on human embryos or on models to see what effects these are. And what we're left with then is oftentimes congenital pediatric disorders and other types of things at early stages. And these are important because these are oftentimes where significant or minor interventions can have significant outcomes over the lifespan of those individuals. and yet we don't know what approaches perhaps to take. So I see this one again as tremendous abilities, not again to grow babies or make things. Again, I think that that is not in the realm or scope of what anyone is trying to do. But if we can use some of these tools that are coming in this pipeline, we might actually be able to start to have some resources, have some platforms, and have an ability to approach these in completely new and novel ways. And I think that's something to me that excites me as well, to think about this and think about how to plug in with folks, obviously in the Atlanta area, like Children's and others, where there's been tremendous interest. And Chun Wee, who will speak today, is actually a recent hire of Children's, where they've invested significantly in their research program to try and really build up in these areas. So I'll stop there for sake of time. I'll take time for one or two quick questions, but I'll be around afterwards, so I'm happy to do that. And then I want to be able to jump to our first speaker, Steve Stice, as quickly as possible. So any quick questions? No? Okay. So then let me go ahead and, like I said, let's keep moving with the session, because we have 4 outstanding talks. That was my last slide. So these are the speakers for, come on up, Steve, it's fine. These are our 4 speakers for this morning. Steve Stice will lead us off as GRA Eminent Scholar from UGA, and probably the most seasoned and experienced stem cell researcher we have in the state of Georgia. Chun Wee Zhu, one of our also highly experienced, actually comes from Geron, and so she probably has more knowledge and familiarity about working with human ES and pluripotent than most people in the world, and now actually resides just across town at Emory. John Temenoff, my colleague and collaborator, who has done a ton of work developing the types of some novel systems where you can actually start to dissect crosstalk between different types of populations. And then Melissa Kinney, who's actually a graduate research student from my lab, will be defending sometime in this coming year, who's done a lot of really cool work with engineering and thinking about how we can start to move towards 3D using 3D pluripotent stem cells in a 3D fashion to start to do some tissue modeling. So I'll stop there.